So here we are looking at the physical factors that affect bacterial growth. So things that can either speed up or slow down the rate at which bacteria grow. And so we'll first get an overview of all the physical factors, and then we'll look up close at pH and temperature and their effects on bacterial growth. So physical factors in an environment include things like pH and the temperature at which the bacteria are being grown, how much oxygen there is in the atmosphere and in the growth medium, the amount of moisture that they're provided to grow on or with, hydrostatic pressure that they may be under, osmotic pressure they may be subjected to, and then the radiation that they may be experiencing. So these are all physical factors that can have pretty significant effects on how well bacteria grow, and will allow us to also, along the way, inhibit bacterial growth by using these physical factors as means of inhibiting their growth. So in this the first of the physical factors we're going to look at is pH, and if you recall from a chemistry section of a anatomy and physiology or some other class that you've had, pH is a measure of the acidity or of the basicness, the alkalinity of a solution. Solutions that are very acidic, they have lots of hydrogen ions that are running around in them, and those hydrogen ions can be really interfere with chemicals, and they can go in there and break up chemical bonds. On the opposite end, basic solutions are ones that bind up hydrogen ions, that will remove hydrogen ions from solution, and they also have the ability to do some pretty significant damage to chemicals. So microorganisms all have an optimum pH at which they grow best. For most microbes, this is somewhere near neutral pH, and if you recall, the pH scale starts down here at 0 and it goes to 14, and neutral is right in the middle at 7. So for most bacteria, neutral pH is where they grow best. And whatever the optimum pH for the bacterium is, usually if you shift more than one pH unit above or below that optimum pH, then you move the bacterium outside of its comfort zone and it won't grow. So pH tolerance, the ability to tolerate a range of pHs, all relates directly to protein function. Because when pH shifts away from whatever the optimum is for that bacterium, then the proteins get denatured, they unfold, and they lose their function. And so primarily what we're talking about is that as pH gets away from optimum, then the functioning of the enzymes that are making chemical reactions happen, and the transport proteins that are found in the membranes of the cells, the function winds up being lost. One thing that can mitigate the effect of pH and can help bacteria tolerate extreme pH, so really basic conditions or really acidic conditions, is the cell wall. So the cell wall can help to prevent the cell membrane from being exposed to the extreme pH. And because bacteria don't like to be outside of whatever their optimum is, most growth media include in them some sort of buffer chemical. A chemical that will either bind hydrogen ions if the solution gets acidic, or will release hydrogen ions if the solution becomes really, really basic. So buffers help to maintain that appropriate optimal pH for the bacteria. And so by putting a buffer in, then you can help ensure that the bacteria don't get pushed outside of that comfort zone. So we can classify bacteria by their requirement for or their tolerance of specific pHs of acidic or alkaline environments. So an acidophilic bacterium, acidophiles, these are ones that grow best at very low pH, between 0.1, that's very acidic, up to 5.4, which is less acidic than, say, lemon juice. Is somewhere around 4, if I remember properly. So one example of an acidophile is lactobacillus, which is a bacteria found in yogurt or any other fermented foods. So sauerkraut, if you make your own sauerkraut, or make your own yogurt, or if you make your own kombucha or whatever that you have to ferment, then you're going to be growing lactobacillus. And then there are these structures that are found in caves that are grow in really acidic environments called snottites. And so these snottites are cultures of bacteria that are growing in very, very acidic sulfuric acid. So lactobacillus prefers a little bit of acidity, but not really extreme. The bacteria that make these snottites, these really slimy kind of stalagmites in caves, they grow on very, very acidic media. Neutrophils, these are the ones that grow best at a pH somewhere between 5.4 to 8. And most human pathogens are neutrophils. They like that moderate pH. They don't want to be in a real extreme pH. And then alkalinophiles are ones that grow best anywhere between 7 to 11.5. So there's a little bit of an overlap there with neutrophiles and alkalinophiles. One example of an alkalinophile is agrobacterium. That's a bacterium that grows in soils that have very, very high pHs, around 12. And here's some agrobacterium here. These little rods is the agrobacterium. And Vibrio cholera, that's the bacterium that causes cholera, which is an intestinal disease. So if you get cholera, you wind up with really, really terrible diarrhea, and then it's spread when contaminated waters are consumed by another person. So here is the Vibrio cholera bacteria, and it grows best at a relatively mildly basic environment, a pH of about 9. So a real range among the bacteria for their tolerance of either acid or base, but most human pathogens are in that neutrophile range, or something like Vibrio, which grows best at just outside of sort of a comfortable human range. Okay, so temperature also plays a big role in how rapidly bacteria grow. And most bacteria, in the same way that bacteria can span, around pH, about 2 pH points, a pH point above and below their optimum. So if this is the optimum pH here, we can go up +1 or down -1. Then there's also a range around which most bacteria grow, and that is about 30 degrees Celsius temperature range around that optimal range. But the maximums, there are some species that can still actually grow at below 0 degrees C. They live in really salty Arctic Ocean waters. And then there are some species that grow at higher than 100 degrees C. So that's hotter than boiling. And so in some of these hot springs where the spring is under pressure and you can get higher than 100 degrees C temperatures, then there are still bacteria that grow at that temperature. So the optimum temperature, again, for each species, just like we see with pH, is totally related to enzyme function as well as the ability of proteins to transport, those transport proteins that are found in the membrane. And we can classify bacteria based on the optimum temperature as well. So psychrophiles, these guys like cold temperatures. Thermophiles, they like it hot. And mesophiles, they like the moderate temperatures. And I bet you can guess which ones of those are going to be most likely to be human pathogens. But I'll let you come closer. We'll look. So psychrophiles, we'll talk about them first. These are ones that grow best at low temperatures, between 15 and 20 degrees C, although some can grow as low as 0 degrees C or even colder than that. None of these, of course, are going to live in the human body. because if you're, the human body is up here at 37 degrees C, so if your optimum temperature is around 15, you've only got about 15 degree points to go up and 15 to go down, so 37 is going to be outside your optimum. Listeria monocytogenes is one example of a psychrophile. Listeriosis is the disease that they cause, and listeria are capable of spoiling even refrigerated foods because they like to be at those cooler temperatures. A refrigerator, I think we keep our refrigerators somewhere around, I forget what it is in Celsius. But if room temperature is 25, then we're going to keep our fridge, I know what it is, about 4 degrees C. That's like the ideal, so slightly slightly warmer than freezing at 0 degrees C. So psychrophiles, they like it cold, 15 to 20 degrees C for an optimum temperature. Obligate psychrophiles are ones that can only grow at very cold temperatures. So for instance, Sporosarcina globosporus, and here is an example or a picture here of Sporosarcina globospora. It cannot grow at temperatures above 20 degrees C. So you warm it up above that, it may survive, but it's not going to be growing and dividing. So obligate psychrophiles must be cold. Facultative psychrophiles, when you see this facultative term, that means they prefer whatever the temperature range is, so they prefer cold temperatures, but they can still grow at warmer temperatures as well. So, facultative just means that they are capable of using or they prefer to use whatever that condition is, but they can live without whatever the condition. So in this case, facultative psychrophiles, they like it cold, but they can still do it at warmer temperatures. And I should have pointed out, here's an image of Listeria monocytogenes, that one that causes listeriosis in food as food spoils. Okay, mesophiles, they grow best at between 25 and 40 degrees C. And so as you can imagine, this is the group that's going to include most of the pathogenic bacteria for humans, because most of our body surfaces are somewhere between room temperature, like at the surface of the skin, and 40 degrees C, which would be core body temperature. 37 is core body temperature. Thermoduric organisms are organisms that are mesophiles, so they have an optimal temperature that's in that 25 to 40 degrees C range, but they can withstand brief exposure to high temperatures. So these are the ones that can spoil canned foods that haven't been fully processed. So if you get some canned green beans, say, from a friend, and you notice that the seal is bulging a little bit, that they have made, these are ones that they canned themselves, then chances are good that one of these thermoduric organisms has withstood the processing that they did to the canned goods, but they didn't process it long enough. So Clostridium botulinum, that's the organism here. It's a rod that produces botulism. And Enterococcus faecalis, it's a gut organism, so we find it in our small intestine, large intestine. But if it gets into other locations, say for instance, somebody isn't wiping their rear properly when after a bowel movement, then a UTI could result from that. Endocarditis, cellulitis, these are all inflammations caused by Enterococcus getting into some place where it doesn't belong. So Enterococcus faecalis, a thermoduric organism that can withstand a little bit of processing. And so an example here would be, maybe back in the day when we reused catheters, perhaps after autoclaving them. If they didn't get autoclaved properly, or if they got contaminated after they've been autoclaved, then, or some other, medical device like that, it could be that you're spreading an infection then of Enterococcus. So mesophiles, the ones that are most likely to be human pathogens, and then the thermophiles are the ones that grow best at very hot temperatures, usually between 50 and 60 degrees C. And so these include the bacteria that we find in our compost piles. So if you have a compost pile and you know how it heats up, well, it heats up, and that is good for the thermophiles. And it could be, say, the bacteria that live in these hot geysers that are found in Nevada. And this is kind of a cool story. So the Fly Ranch is the name of the location of this. So it's a privately owned ranch, and back in the 60s, some speculators went in there trying to drill for oil. They instead wound up tapping into a hot spring, and this is what's developed, this really gorgeous, like mountain landscapey looking thing with the runoff forming these really crazy pools. And in the pools you find different species of sulfur bacteria. The ones that can withstand the hottest heat are right up close to where the geyser is spouting. The ones that can tolerate can't tolerate the really hot heats, but can tolerate really warm temperatures still, would be living in the pools of the runoff of that. So obligate thermophiles are ones that they are obligated to grow at really warm temperatures, so above body temperature. Again, a facultative thermophile is one that prefers to grow above 37, but it can grow at lower temperatures. And so Bacillus styrothermophilus, try to say that one, is one of these facultative thermophiles that can cause food storage. So it prefers to be at really warm temperatures, but as the food cools, if there's a bacillus population that gets introduced somehow, then you can still get it to grow if you leave it sitting out on the counter, for instance, a hot food that should have been kept hot but isn't. All right, so those are the thermophiles. So critical growth temperature is, did I go too far? No, okay, I guess I must be, let me just check my notes here before I go on. Okay, so the critical growth, the growth temperatures, the preferred temperatures of bacteria are the result, once again, of enzyme function. So for every 10-degree increase in temperature, enzyme activity doubles until you reach the temperature that the enzyme denatures and chemical reactions can't occur. On the other hand, if you cool down bacteria for every 10 degrees C decrease in temperature, then enzyme activity halves. So let's say we have a bacteria that can tolerate a range of maybe 25 to 45. Its preferred temperature is, let's say, 35, so close to body temperature. So if we reduce the temperature at which that bacteria is being stored or kept by 10 degrees, then you will have half the growth after 24 hours that you would see in a 35-degree temperature culture of that bacteria. So where does this become important? Well, it becomes important around holding hot and refrigerated foods at appropriate temperatures. So by holding hot foods at about 63 degrees C, then we can reduce the growth rate of common thermophiles that might infect the food and cause it to spoil. And it will generally prevent growth at 63 C. It'll prevent the growth of most other bacteria. Refrigerating food to 4 degrees C, that's going to slow the rate of most psychrophiles to prevent their ability to spoil the food, and it prevents the growth of any mesophilic bacteria. And then freezing food down to minus 30, that will stop the growth of even the psychrophiles. So these are the temperatures that we are required, that restaurants and other food service groups are required to hold temperatures or hold food at. So if you're trying to keep a food hot, you have to keep it at 63, give or take. If you're trying to keep a food cold, then it needs to be kept at 4. And if you're trying to preserve a food for long term, then you want to keep it at less than minus 30 degrees C in order to do that. So these are three. That's what happened. I got my slides out of order. I need to fix that. So here's the first slide that was supposed to go in front of that one. There are three critical growth temperatures that can be determined then for most bacteria species. The first is the minimum growth temperature. So for typical thermophiles, let's look here, the minimum growth temperature is somewhere around 43 degrees C for this thermos the thermos genus of bacteria. The maximum growth temperature for the typical thermophile is somewhere around 69 degrees C. But here at the top of the peak, that is where we see the most rapid growth rate of a thermophile. And that's somewhere around 60 degrees C is the most rapid rate. And then we see as we get up above 60 degrees C, we see a very rapid drop-off. And that drop-off is so rapid because all of the enzymes are denaturing and chemical reactions are stopping once we get past that optimal growth temperature. So here's where we see the slowing down of the bacterial growth, with every 10 degrees typically about halving bacterial growth. Okay, so there you have it. pH and temperature and their importance in determining how rapidly bacteria can grow and divide. You know what to do.